The aim of this procedure is to determine the force generated and fatigability of isolated skeletal muscle as a way to quantitate the effects of genetic modification or therapy. First, the extensor digitorum longus muscle is dissected from the mouse hind limb. Then, the muscle is mounted in a muscle strip myograph. The next step is to ascertain the voltage for stimulation and pretension of the muscle at which maximal twitch tension is achieved. The final step is to determine the force-frequency relationship for the muscle, as well as the onset and degree of fatigue with low-frequency tetanic stimulation. Ultimately, results can be obtained that show measurable changes in myomechanical properties of the muscle through myography.
The main advantage of this technique over existing methods, like exercise testing, is that myography allows for a quantitative assessment of muscle strength and function. This method can help answer key questions in the muscle biology and muscular dystrophy field, such as whether specific therapies or genetic modifications alter muscle function. Generally, individuals new to this method will struggle because rapid, atraumatic dissection of the hind limb muscles takes practice and acquired skill.
We first adopted this method when we realized that quantitative measures for assessing muscle function are needed, as new cell and bioengineering-based therapies for muscle disease are tested in preclinical animal models. Visual demonstration of this method is critical, as the steps needed to properly handle the muscle are difficult to master. Demonstrating the procedure will be Wenhui Gong, a research scientist, and Sampab Cholseri-Punlert, a graduate student in our laboratories.
Following euthanasia of a mouse, arrange the carcass ventral side up on a dissection tray and pin the leg to the tray. Under a dissection microscope, cut open the skin and carefully open the fascia. Next, peel the tibialis anterior muscle from the ankle upwards to expose the extensor digitorum longus, or EDL muscle. Use drops of lactated Ringer's solution to keep the muscle moist during the harvest. Then dissect out the EDL, preserving as much tendon as possible on each end and being careful not to touch the EDL muscle fibers themselves. Put the EDL muscle into a petri dish containing lactated Ringer's solution. Now tie sutures to each of the muscle tendons. These studies use a tissue bath to hold the muscle and a force transducer to measure muscle tension. In addition, a square pulse electrical stimulator and data acquisition platform are used to elicit, record, and analyze myomechanical responses. The next step is to fill the myograph bath with 6.5 ml of Krebs Henselite solution. Warm the bath to 25 degrees Celsius. Bubble the Krebs solution with 95% oxygen, 5% carbon dioxide. After bubbling for 15 minutes, use sutures on the tendons to transfer the EDL muscle to the bath. Secure the tendons between the clamps of the myograph. Be careful not to clamp the muscle itself. Maintain the myograph bath at 25 degrees Celsius. To begin the tension analysis, adjust the initial muscle length so that it is equal to the in situ muscle length. Using a stimulus duration of 0.5 milliseconds, gradually increase the voltage to determine the stimulus required to obtain maximal twitch tension. Then, set the stimulus 20% higher to achieve a supramaximal stimulus, which is usually about 40 volts. The next step is to gradually stretch the muscle until there is no further increase in twitch tension. This length is considered the optimal muscle length. Allow the muscle to equilibrate for 3 minutes. Then, keeping the muscle at its optimal length, deliver a supramaximal square stimulus and record the output. Record the twitch tension curve. In this figure, the maximal twitch tension is labelled PT. The contraction time is labelled CT. The half relaxation time is labelled HRT. The bar represents 1 second. Following a 3-minute rest period, Measure the tetanic tension by applying a strain of supramaximal stimuli for 300 milliseconds at 150 Hz at optimal length. This procedure generates a tetanus tension curve. The maximal tetanic tension is labelled PO. The half relaxation tetanic tension is labelled HRTT. The bar represents 1 second. After a 3-minute rest period, begin the force frequency analysis by applying 300 millisecond trains of supramaximal stimuli at 30, 60, 100, 140 and 160 Hz. Allow 3 minutes of rest between each train of stimuli. After a 3-minute rest period, begin the fatigue analysis by applying trains of short tetani. 60 Hz for 300 milliseconds every 3 seconds for 10 minutes. By 10 minutes, the tetanic force should decline to a plateau level of around 15% of the initial value. After finishing the force recordings with the muscle at optimal length, measure the muscle diameter using the ocular on the microscope. Following this, remove the sutures and weigh the muscle to determine muscle mass. The last step is to weigh the mouse to assess body mass. These measurements are used for the calculations listed in the accompanying article. This figure shows the increase in tension generated with increasing stimulus frequencies. Shown here is the muscle's response to a pulse train at 30 Hz. This figure shows the greater force elicited at a pulse train of 140 Hz. This graph shows the force-frequency relationship plotted as the percent maximal force versus the stimulation frequency. The shape of the force-frequency curve is characteristic of muscle strength and can be used to make comparisons between muscles from different animals. This figure demonstrates muscle fatigue over the course of 10 minutes of low-frequency stimulation. These figures show the muscle's response to the stimulus train at the indicated time points. This plot shows the low-frequency muscle fatigue as a plot of percent maximal force versus time. The shape of the low-frequency fatigue curve is characteristic of muscle strength and can be used to make comparisons between muscles from different animals.
Once mastered, this technique can be done in 30 minutes. While attempting this procedure, it's important to remember to have all of your equipment ready to minimize the time between muscle harvest and analysis.
Following this procedure, other methods like immunohistochemistry, Western blot, and quantitative PCR can be performed in order to answer additional questions, like whether and where specific myogenic proteins and stem cell markers are being expressed in the treated muscle. After watching this video, you should have a good understanding of how to dissect the muscle hind limb, mount the muscle in a muscle strip myograph, such as the one we have shown you from Danish Myotechnology, and measure changes in myomechanical properties.